Welcome to BioBits and Bytes. Today, we're diving deep into a really powerful set of tools, things biologists and medical folks use all the time. We're talking blotting techniques and ELISA,
Hmm.
basically ways to look at the fundamental building blocks of life DNA, RNA, proteins. Our mission really is to unpack these, figure out what they are, but also, why they're so crucial.
Absolutely. And we've got some great source material today, haven't we? Guides from Bolster Bio, PubMed, Life in the Lab, some SlideShare stuff, Wikipedia, even a research paper from the Future Journal of Pharmaceutical Sciences. So, yeah, plenty to cover the principles, the procedures, the applications, southern, northern, western blotting, and ELISA too.
Fantastic. And we're really glad to have you here to guide us through it all. So let's just jump straight in. For listeners maybe not so familiar, what exactly are these blotting techniques? Can you give us a definition, sort of the core idea?
Sure. So, um, at its heart, blotting is about taking biomolecules, DNA, RNA, proteins that you've already separated, usually by size, on a gel,
Mm-hmm.
and transferring them onto a different surface, typically a membrane. This membrane kind of holds them in place, immobilizes them right where they landed on the gel. And the key thing is, once they're on that stable membrane, you can use specific probes to find exactly what you're looking for.
Probes,
Like labeled bits of DNA or antibodies, things that will stick only to the specific molecule you want to identify. It's like copying info onto sturdier paper so you can search it easily.
oh, okay, that makes sense. Transfer and detect. Now we hear these names, southern, northern, western blotting. Sounds a bit like a joke almost. Can you walk us through each one? What molecule does it look at? What's it used for? Let's start with southern.
Right, Southern blotting. It's named after Edwin Southern, the scientist who developed it. And this one is all about DNA. Its purpose is analyzing and identifying specific DNA sequences. The process usually starts by taking your DNA sample and chopping it up. Yeah, using special enzymes, restriction enzymes. Think of them as tiny molecular scissors that cut DNA only at very specific points.
Okay, so you get a unique set of DNA pieces, like a fingerprint.
Exactly. You get these DNA fragments of different lengths. Then you separate these fragments based on how long they are. We use a method called agarose gel electrophoresis. It's like, imagine pushing the fragments through a gel mesh. The smaller pieces wiggle through faster and travel further. The bigger ones get tangled up more, move slower.
Creates a pattern of bands then.
Precisely. A distinct pattern based on size. Now, DNA in the cell is double-stranded, like a ladder. Before transferring, you treat the gel with an alkali solution. This denatures the DNA, basically. It separates the two strands of the ladder
I do that.
so that your probes, which are single-stranded, can actually find and bind to their matching sequence on the DNA strand. Then comes the blotting part, transferring these single-stranded DNA fragments from that delicate gel onto a solid membrane, usually nylon or nitrocellulose.
Right, so you've moved the DNA pattern onto something more durable. How do you make sure it actually sticks there?
Good question. You immobilize it, usually by baking the membrane or hitting it with UV light. That fixes the DNA onto the surface. Now it's ready for detection. You introduce labeled probes.
The things that find the specific sequence.
Exactly. These are single strands of DNA or RNA designed to be complementary to the target sequence you're hunting for. And they're tagged maybe with a radioactive atom or a fluorescent dye or something that glows chemically.
And these tags then, they hunt down their matching DNA on the membrane.
They do. You give them time to hybridize. That's the scientific term for binding to their complementary sequence.
Yeah.
Then you wash away any probes that didn't find a match. And finally, you visualize where the probes did bind. If it was a radioactive probe, you might use X-ray film, kind of like taking an X-ray picture. If it was fluorescence or chemiluminescence, you use special cameras. That tells you where your target DNA sequence is.
Wow. Okay, southern blotting is quite a detailed process. What are the main uses? Why go to all that trouble?
It's incredibly useful. You can see if a specific DNA sequence is even present in your sample. You can study DNA methylation chemical tags on DNA that affect gene activity. That's important in, say, cancer research. It's also the basis for RFLP analysis, restriction fragment length polymorphism. That was huge for genetic fingerprinting, in forensics and paternity testing.
Right, I've heard of that.
Yeah. Plus, identifying gene mutations, variations between individuals, mapping where genes are on chromosomes, cloning genes, figuring out the size of DNA pieces, how many copies of a gene someone has, lots of applications. Of course, you need the right kit. Those restriction enzymes, the gel setup, buffers, the blotting machine, an oven for hybridization, and crucially, those labeled probes.
We've got an image here too showing the gel bands and then the probes lighting up on the membrane. Helps visualize it. Okay, let's move north then. Northern blotting. The name suggests a link, but this is for RNA, right?
You got it. Northern blotting follows the same kind of logic as southern, but the target molecule is RNA. So first step, get the RNA out of your cells or tissue. Often, researchers are specifically interested in messenger RNA, or mRNA, because that's the molecule carrying the instructions from DNA to make proteins.
So it tells you which genes are active.
Exactly. And for eukaryotes, organisms like us, mRNA, often has a specific tail, a poly-A tail. So you can use a neat trick with tiny DNA pieces called oligo-DT that stick to that tail and basically fish out the mRNA from the total RNA pool.
Clever. Okay, so you've got your RNA, maybe mostly mRNA, then separate by size again.
Yep, separate by size using gel electrophoresis. But RNA is a bit different from DNA. It's single-stranded and can fold up on itself. To make sure it separates just by length, you need to keep it unfolded. So the gel usually contains denaturing agents like formaldehyde in an agarose gel or urea in a polyacrylamide gel. Keeps the RNA strands straight.
And then, just like southern, transfer to a membrane.
Correct. Transfer the separated RNA onto a membrane. For northern, positively charged nylon membranes work really well because RNA is negatively charged, so they stick together nicely. After the transfer, you immobilize the RNA, again usually with UV light or heat.
And detection. Still using probes.
Still using probes. Labeled nucleic acid probes can be DNA, RNA, or short synthetic bits called oligonucleotides. They need to be complementary to the RNA sequence you want to find, and again, you label them. Radioactive labels like phosphorus-32 were common, but now non-radioactive methods using chemiluminescence are very popular too. They use enzymes like alkaline phosphatase or horseradish peroxidase attached to the probe.
HRP. Right, I've seen that.
Yeah, HRP or AP. Then you detect the signal, maybe with X-ray film or a digital imager, using densitometry to measure how strong the signal is, how dark the band is.
Okay. Now, getting that probe to stick correctly, it seems like things could affect that.
Oh, definitely. Hybridization is sensitive. Things like the salt concentration in your buffer, how thick or viscous the solution is, how long the probe and target sequence are, how many mismatches there are between them. Even just the ratio of different bases, G's and C's versus A's and T's, can affect how strongly they bind. You have to optimize the conditions.
So what are the big plus points for northern blotting? And any major drawbacks?
Well, a key advantage is determining the size of the RNA. That can tell you a lot. It's also good for spotting alternative splice products, where one gene can actually produce slightly different versions of RNA, leading to different proteins. You can even use probes that aren't a perfect match sometimes to find related RNA sequences.
Okay.
Plus, you can check your RNA quality on the gel before blotting, and the membranes can be stored and reprobed later, looking for other RNAs. And like I said, non-radioactive methods are often just as sensitive now. Some argue it can pick up subtle changes in gene expression better than, say, microarrays in certain contexts.
But the downsides?
The big one is RNA itself. It's really fragile. Enzymes called RNases are everywhere, and they love chewing up RNA, so you have to be super careful, use sterile techniques, special inhibitors.
Right, RNAs free everything.
Exactly. Also, some chemicals used are hazardous formaldehyde, radioactive stuff if you use it UV light, and generally it's less sensitive than a technique like RT-PCR, which can detect tiny amounts of RNA. But Northern offers good specificity, and that size information PCR doesn't give you directly.
So where would you typically see northern blotting being used most?
Primarily for looking at gene expression levels. How much RNA from a specific gene is present? Does it change under different conditions? Also, analyzing the size of that RNA and detecting those different variants, like splice variants, gives you a really solid picture of a specific RNA transcript.
Right. We've covered DNA with southern, RNA with northern. Time to head west, I guess. Western blotting. Exactly. Western blotting, or immunoblotting, as it's also called. This one is the powerhouse for looking at proteins. And yes, the name was definitely a bit of fun, following the southern and northern theme, coined in the late 70s, sort of tongue-in-cheek.
But the focus here is entirely proteins.
Entirely proteins.
Does the basic idea separate, transfer, detect still apply?
It does, absolutely. You start by separating your proteins, usually by size. The standard method is SDS-PAGE. That stands for sodium dodecyl sulfate polyacrylamide gel electrophoresis.
Bit of a mouthful.
Aha, yeah. Basically, SDS is a detergent. It unfolds the proteins and coats them all in a negative charge. So when you apply electricity, they move through the polyacrylamide gel pretty much just based on their size. Smaller ones go faster, bigger ones slower.
Just like with DNA and RNA then?
Similar principle, yes. Once they're separated in the gel, you transfer them to a membrane. For proteins, it's typically PVDF or nitrocellulose.
Okay, proteins transferred. Now, how do you find your specific protein of interest among all the others on that membrane? This is where antibodies come in. It's usually a two-step antibody process.
Right.
First, you use a primary antibody. This antibody is chosen because it specifically recognizes and binds only to your target protein. It's like a highly specific key for that protein lock.
Okay.
After letting that bind, you wash away any unbound primary antibody. Then you add a secondary antibody. Now, this secondary antibody doesn't bind to your protein directly. Instead, it's designed to recognize and bind to the primary antibody.
Ah, so it sticks to the first antibody.
Precisely. And crucially, this secondary antibody has something attached to it, usually an enzyme like HRP, horseradish peroxidase, or maybe a fluorescent tag.
So the secondary antibody brings the signal, like a signal flare attached to the first antibody.
Exactly. It amplifies the signal. If it has an enzyme like HRP attached, you add a chemical substrate. The enzyme reacts with the substrate and produces light that's chemiluminescence, or maybe a colored spot. If it's a fluorescent tag, you shine a specific wavelength of light on it, and it glows. You see a band appear on the membrane at the correct molecular weight for your protein, and bingo, you know it's there.
That's really quite elegant. What sort of information does a Western blot give you?
Well, obviously, it tells you if your protein is present or not, but also how much of it is there. You can estimate expression levels. You confirm its molecular weight. You can even see if the protein has been modified in some way, like phosphorylation. There are variations, too, like far Western blotting, which can actually look at protein interactions. It's fundamental in research, but also used diagnostically for HIV confirmation, testing for mad cow disease, sometimes Lyme disease, too.
And you mentioned it's evolved. Advancements in the technique.
Oh, yes. Things like single-cell Western blotting. Now that researchers look at proteins in just one cell at a time. Huge for studying mixed populations. There are faster ways to do the transfer step, more efficient transfer methods. Ways to look at multiple proteins on the same blot, semi-automated systems to make it easier. And using molecular weight markers, kind of like a protein-sized ruler run alongside your samples, is absolutely standard practice, essential for knowing you're looking at the right band.
The sources also mention loading controls. Why are they so important?
Ah, yes. Loading controls are vital if you want to accurately compare protein levels between different samples. You need to be sure you loaded the same total amount of protein in each lane of your gel to begin with. Otherwise, comparing band intensities is meaningless.
Right, could just be you loaded less samples.
Exactly. So you typically probe for a housekeeping protein, something like beta-actin or tubulin, structural proteins that are usually present at constant levels in all your cells, regardless of your experiment. You use the signal from this control protein to normalize the signal from your target protein. Alternatively, you can stain the whole membrane to see the total protein in each lane using stains like Kumasi blue or newer stain-free technologies to confirm equal loading.
In presenting these results, how do researchers usually show them?
They'll typically process the image of the blot using software. Might be specialized stuff like Sugo or just image editors like Illustrator or PowerPoint. To align the lanes properly, maybe adjust contrast slightly to make bands clear and create figures for papers or presentations.
Any major pitfalls or limitations with Westerns?
Well, like any technique, it requires care. Poor technique can definitely lead to inaccurate or misleading results. Things like choosing the right antibodies, optimizing the blocking and washing steps, it all matters.
Right.
It often takes a bit of optimization to get a clean, specific signal for your particular protein.
Okay, we've done the tour, Southern, Northern, Western. Let's switch tracks a bit to ELISA. How does that fit in? How does it compare? And what's it mainly used for?
Right, ELISA. Enzyme-linked immunosorbent assay. It's another type of immunoassay, meaning it uses antibodies. But the format is quite different from blotting. ELISA is usually done in 96-well plastic plates. These plates are designed so that antibodies, or antigens, the molecules antibodies bind to, can stick to the surface of the wells.
96 wells, so you can test lots of samples at once.
Exactly. That's a major advantage. It's very amenable to high-throughput screening.
What's the basic workflow for an ELISA?
It generally involves a few core steps. First, you coat the wells of the plate. You might coat them with an antibody if you're trying to capture an antigen from your sample, or coat them with an antigen if you're trying to detect an antibody. Then you block the remaining surface of the well, usually with a protein like BSA, to prevent things sticking non-specifically later.
Stop background noise, basically.
Precisely. Then you add your sample. If the target molecule is there, it'll bind to whatever you coated the well with. Then comes washing. Washing is crucial in ELISA. You wash between pretty much every step to remove anything that hasn't bound specifically. Next, you add a detection antibody, which is linked to an enzyme. Finally, you add the enzyme substrate. The enzyme converts the substrate into a colored product, or maybe a fluorescent or chemiluminescent signal. You measure that signal, usually with a plate reader, like a spectrophotometer. The strength of the signal tells you how much of your target molecule was in the sample.
You mentioned antigens and antibodies as key reagents. Just a quick refresher for us.
Sure, an antigen is any substance, usually a protein or carbohydrate, that triggers an immune response specifically, the production of antibodies against it. Antibodies are the proteins the immune system makes to recognize and neutralize that specific antigen. They bind very tightly and specifically. ELISA totally relies on this specific antigen-antibody binding.
And there are different flavors of ELISA: direct, indirect, sandwich, competitive.
Yes, those are the main types, depending on exactly how you set it up and what you want to detect. In direct ELISA, the plate is coated with antigen, and you use a primary antibody directly linked to an enzyme to detect if that antibody is present in your sample. Simple, but requires a labeled primary for each target. In indirect ELISA, you also coat with antigen. You add your sample, maybe looking for antibodies in serum. Then you add an unlabeled primary antibody, followed by an enzyme-linked secondary antibody that detects the primary. More sensitive than direct and more flexible as one labeled secondary can work with many primaries.
Okay.
Sandwich ELISA is probably the most common for detecting an antigen. You coat the plate with a capture antibody, add your sample, the antigen gets captured. Then you add a second detection antibody that binds to a different spot on the captured antigen. This detection antibody is either enzyme-linked itself or detected by an enzyme-linked tertiary antibody, like a sandwich antigen antibody, for significant sensitivity.
Right, trapping the target.
Exactly. And competitive ELISA is a bit different. It's often used to measure the concentration of an antigen or antibody. Essentially, the molecule in your sample competes with a labeled version of the same molecule for binding sites on the plate. The more target you have in your sample, the less signal you get from the labeled competitor.
Interesting. So, comparing ELISA to blotting, what are the pros and cons?
Well, ELISA is generally faster and simpler for just determining if a protein is present and for quantifying it. Especially if you have many samples, that 96-well format is great. Sample prep is often minimal, and it can be incredibly sensitive, detecting very low concentrations. You can also get relatively inexpensive kits, especially if you're set up to coat your own plates. Some procedures might have a few extra steps, like indirect or sandwich assays. And critically, ELISA doesn't give you information about the size of the protein, unlike Western blotting. If there are different forms or fragments, ELISA might detect them all without distinguishing them. Also, important to note, while ELISA is widely used in diagnostics, many kits sold are labeled for research use only, RUO, and haven't gone through the validation needed for clinical diagnosis.
It's just amazing the variety of these tools. Looking at the big picture then, why are blotting and ELISA just so foundational in molecular biology and biochemistry?
They're fundamental because they give us windows into the molecules of life. They let us ask very specific questions about DNA, RNA, and proteins. Are they there? How much is there? What size are they? Are they interacting? This allows researchers to piece together how cells work, how diseases develop, how genes are controlled. Each technique has its niche, its strengths. Southern for DNA structure and mapping, Northern for gene expression via RNA, Western for protein levels and modifications. ELISA for high-throughput quantification and detection. They're used everywhere: genetics, cancer research, infectious disease, drug development, you name it. They underpin so much of our molecular understanding.
Okay, let's try and wrap this up. We've covered a lot of ground. Blotting techniques: Southern for DNA, Northern for RNA, Western for proteins. And ELISA, this versatile plate-based assay. They all give us these incredibly powerful ways to investigate the molecules driving life itself. From finding one specific gene sequence to measuring tiny amounts of protein, there's core methods.
And it's worth thinking about how directly this impacts us. These aren't just abstract lab techniques. They're used in hospitals for diagnosing diseases, in labs developing the next generation of medicines, even in forensics. Every single band you see on a blot image or a color changed in an ELISA, well, it's telling a story. It's revealing something about the biology happening at the molecular level.
So really, understanding these techniques gives you a lens to view that complex molecular world. The procedures might seem complicated, maybe a bit daunting at first glance, but the underlying ideas separating things out, then using something specific to find your target, are actually pretty elegant, pretty powerful.
Which brings us to a final thought for you, the listener, to maybe ponder. We're seeing amazing new technologies emerging all the time, things like next-gen sequencing, mass spectrometry, single-molecule imaging. So how do you think these classic techniques, blotting and ELISA, will fit into the future? Will they eventually be replaced? Or will they continue to play a vital, perhaps complementary, role alongside the newer tech? It's something to think about.
Definitely food for thought. Thank you so much for taking this deep dive with us today into blotting and ELISA. We really hope this has shed some light on these essential life science tools for everyone listening.
My pleasure. Thank you.